So we've just talked about three mechanisms by which bacteria can acquire foreign DNA molecules. Now, in general, there is a potential for these nucleic acids to enhance the survival of these bacteria. But it is also possible that that DNA is actually hazardous. So, for example, we talked about viral infection of cells. This is foreign DNA that is going to be hazardous to the cell. And any other DNA that is acquired from the environment or conjugation may also be hazardous to that bacterial cell. So in order to combat this hazardous DNA, bacteria have mechanisms to target and degrade that foreign DNA. So this foreign DNA might reduce fitness. It might actually be detrimental to the cell, as in virus infection. But there's also metabolic costs that are associated with maintaining that foreign DNA. So if it's not beneficial for that bacterium to have that DNA, there's no reason to maintain it because replication and transcription are costly, and also just sort of maintaining the integrity of the DNA, repairing damages, making sure there's no mutations after replication, that all is metabolically or energetically expensive for the cell. So in order to combat this genome infection, essentially, there are several mechanisms that bacteria can use. We've talked about one before, restriction endonucleases, and we've also mentioned probably earlier in the course, CRISPR. May or may not have come up depending on which course you're in. But these two, CRISPR and restriction enzymes or restriction endonucleases, are sequence specific. So the cell identifies specific sequences that have entered the bacterial genome and degrades them. There's also non-specific degradation. This is where the cell simply recognizes foreign DNA and breaks it down. So we'll talk about those two different types. There's a fantastic CRISPR video that I have linked here. It should also be linked on Blackboard under the Chapter 11 file folder. I would like you to watch that. You can either watch that now or you can watch it after this video. It doesn't matter. But it's a really good explanation of how CRISPR works. So we're going to start with CRISPR, which is one of our sequence-specific degradation tools. So a way to block foreign DNA from invading the cell and integrating into the genome or simply being expressed by that cell. So CRISPR is basically cutting-edge biotech. Jennifer Doudna and her associates in 2020 just won the Nobel Prize, or one of the Nobel Prizes for this technique. So CRISPR is basically being utilized as a gene editing tool in non-bacterial systems. But it is a bacterial defense mechanism that has been sort of commandeered by us to use for research purposes, medicinal purposes, etc. So CRISPR, which is our editing tool, stands for clustered, regularly interspaced, short palindromic repeats. So we'll talk about what all of that means in just a minute. So in bacteria, it is an immune response for essentially remembering previous infection and attacking it, sort of like our immune response. But we have developed this as a system for editing DNA. Now, this technique, this CRISPR system, enables researchers to modify any gene in an organism. Now, this could be for knockout purposes. So knockouts are genes that have been inactivated, and usually you would do this to sort of figure out what that gene does. So there are different ways you can knock out genes. CRISPR is not the only way. Essentially, you introduce a mutation into a gene, and then you look at those mutant organisms and see what happens. You can also use this as an editing technique. So to actually change the sequence, let's say you have a dysfunctional allele that causes a disease or that causes an undesirable phenotype. You might want to edit that gene with the quote unquote correct sequence, the one that is not detrimental to the organism. But first, before we talk about that, let's talk about where CRISPR comes from. CRISPR was isolated in bacteria, and as I said, it's essentially an immune response that acts as a mechanism for remembering viral DNA infection. And when it remembers or registers that viral DNA infection, it protects against future infections. It allows the bacteria to recognize more quickly that it is being invaded and attack it, just like your immune system. Your immune system has a memory. You are exposed to something like chickenpox or measles. Maybe it was via vaccine, maybe it was via actually having the disease, and your immune system remembers those diseases. And so when you're exposed again, it can fight it off that much faster. This is how the flu vaccine works. So your body is inoculated against the flu. For one season, it remembers the flu. So if you're exposed to one of the strains of flu in your flu vaccine, you're either not going to get the flu or your body's going to make an immune response much more quickly. And so the effects of the flu are not so great. So CRISPR works in a similar way. Human immune system is a much different system, but the overall idea is the same. It's a way to remember a viral infection. So let's talk about the structure of CRISPR. So in bacteria, there was this set of sequence that contained these repeat sequences where there were repeats followed by spacers, and each of the spacers was different, but all of these repeat sequences were the same. So they found that this CRISPR, this clustered, regularly interspersed, so things in between, short palindromic repeats. So palindromic means that it's the same forward and backwards. So these repeats are palindromes, and they are interspersed in between these spacer sequences. Now, it turns out that these spacer sequences are actually fragments of viral genomes. And this CRISPR sequence is preceded by this group of Cas genes. So these Cas genes are generally nucleases, and a nuclease is an enzyme that cuts DNA. So essentially what's happening here is that this CRISPR system is going to recognize a viral genome. It's going to cut it with this encoded nuclease. And it's going to insert a small chunk of that viral genome in this spacer. So we basically have a library of previous viral infection incorporated into the bacterial genome. So now what's also interesting is because of those palindromic sequences, these palindromes actually produce a hairpin structure. So this is a unique feature of this region. So we've got this Cas system that works alongside this CRISPR system where we're going to cut up DNA that we have recognized as foreign. So here's a drawing from our book that shows this process. So we have viral infection. That virus is going to inject its DNA. So you might remember from our transformation lecture, this is something similar to, or actually the T4 bacteriophage. with its unique shape. We've already talked about that. It will inject its DNA into the bacterium. Now, if this bacteriophage causes an infection, it's going to take over molecular machinery, use that machinery to make new viruses. It's going to lyse, and then these viruses are going to infect other cells. But some of those bacteria are actually going to survive. They're going to recognize that this DNA has been inserted, and they're going to mount a defense against it. And if they survive, and this DNA is cut up, it can then be incorporated into a new spacer, so that the next time one of these bacteriophages infects the cell, can recognize it much more quickly. This DNA here is going to act as sort of a guide for these Cas genes to recognize that viral infection and cut it much more quickly. So once that spacer is inserted into this palindromic repeat section, it's now part of the genome. So this new short RNA sequence can recognize this infecting phage DNA and act as that guide for that Cas9 enzyme, which can bind here and cleave our phage DNA. So that foreign DNA has entered the cell. It's been incorporated in as a sequence in our CRISPR sequence region. And we are going to use that region to make a double-stranded DNA molecule. So now, if this region is transcribed, it can be used to recognize that foreign nucleic acid that's come in, that foreign viral DNA or foreign DNA from another source, and actually recognize the complementary sequence to that original virus that infected it. So let's look at how CRISPR works as a gene editing system. So again, this is a picture from my Bio 100 book. I really like this picture because it's nice and simple. In a laboratory, we can use CRISPR in one of two ways. We can use CRISPR to knock out or to disable a gene, or we can use it to edit a gene sequence, to put in a corrected sequence or a sequence that we want. So we have our gene of interest. Let's say that we want to insert a DNA sequence from, let's say, like a corrected DNA sequence. So let's say that we have a disease that is caused by a mutation, and we want to correct this mutation. So one of the early experiments that was done with CRISPR was an experiment in which they took mice that had a, excuse me, I'm blanking on what it was. Give me just a second. Sorry, that had liver disease. And so they took the corrected gene for this liver disease. They took the sequence from it and made an RNA molecule that was going to act as a guide for that target sequence. And then they took this RNA guide and they inserted it into a plasmid along with the Cas9 sequence. So Cas9 is one of the specific Cas gene sets that is used to recognize and cut the DNA. So this RNA sequence that is very, very similar to the actual gene and in those mice that's causing the disease recognizes the DNA sequence. And the Cas system, which is a helicase plus nuclease, so it opens up the DNA and then cuts it. So it opens up the DNA at the region where this RNA sequence recognizes the complementary DNA sequence of our gene of interest. It actually cuts that sequence in the mouse or whatever organism you're working on. And then a new sequence, or the corrected sequence, can be inserted. Now, the first time this was done successfully in those mice, they were actually able to get this into enough of the liver cells that it actually didn't remove liver disease in these mice, but it actually was able to alleviate the symptoms enough that the mice basically had sort of normal function again. If you're going to use this as a knockout, you would still use your RNA guide, and you would allow your Cas9 system to cut your DNA. So then you have this broken piece of DNA. And then in an attempt to fix this, because our DNA is always making repairs, always checking itself, in an attempt to fix this, the DNA mechanisms in the cell, the DNA repair mechanisms, are actually going to sort of quote unquote heal this, and in doing this add some genes to make a mutation. And so this new sequence then, instead of inserting, or I'm sorry, this region, instead of inserting a new sequence, we're actually going to get this mutation inserted, which is going to produce the knockout. So there's a huge potential for CRISPR. These are just a couple of examples here of things that have already been done. I gave you the mouse example. I guarantee that since this book was published, if you're watching this at any point after I record it, there's plenty of new examples of things that CRISPR has done. But it is a huge breakthrough in the ability to very specifically make alterations or edits to particular genes. So going back to our book, this is an image from Toolbox 11.3. This shows just a couple of examples of CRISPR gene editing. So here in the C. elegans, this little nematode, we have editing of a body shape gene. You can see we've got the normal here, and then we've got this smaller edited nematode with a shortened body structure. Here we have a gene that affects the shape of the zebrafish embryo, so it's affecting development of embryos. And we also have an eye color change here. This red eye is our normal, and then we've got this sort of sepia or brown colored mutant gene for this fruit fly. So these are just some examples of how this could be used in a laboratory setting. All right, so our other mechanism that specifically recognizes invading sequences are restriction endonuclease systems, or restriction enzyme modification systems. So a restriction endonuclease is going to recognize and cut a short specific sequence of DNA. So if you're watching this in fall 2020, we've already done this lab. If you're watching it later, after fall 2020, we may not have done this lab yet. If not, no worries, we're going to do it. So as an example here, we've got EcoR1. This is a restriction enzyme that was isolated from E. coli. They've been isolated from many different types of bacterium. There's tens, maybe even 100 different types of restriction enzymes. And these restriction enzymes are basically an immune response. So in bacterial systems, they recognize these sequences and they cut up foreign DNA with those sequences. So here we've got the EcoR1 sequence GAATTC. So anywhere the cell sees the enzyme recognizes this GAATTC and only that sequence, it is going to make a cut. Now this specific enzyme cuts between the G and the A and releases this the bonds between the complementary strands, and it cuts on both sides. So in this direction, GAATTC, it recognizes and cuts between the G and the A. On this opposite strand, we have a palindrome. So the palindrome part of this restriction enzyme system is really important because it allows the system to cut both sides at the same spot. And that leaves us with this sticky end. Now these guys could come back together. If we allowed them to hang out, they would come back together because they are complementary. Now this is important. We already talked about plasmids and transformation. So we can use this in a lab by cutting two pieces of DNA from different systems. Let's say we have our plasmid and our gene of interest. We talked about this in an earlier lecture, earlier lecture for this chapter. Then we can allow them to combine. So we can insert our gene of interest into our plasmid. And the reason that this works is because of these palindromic sequences with sticky ends. Now, you might be thinking, well, how does E. coli not cut its own genome with EcoR1? Well, it doesn't cut its own genome because every organism makes specific modifications to its own genome. EcoR1, when it performs its DNA replication, it actually does some what we call methylation. This is a methyl group, the CH3, and it adds methyl groups to some of its adenines. So if EcoR1 finds this sequence GAATTC, but it's methylated because it's in its own system, it won't cut at that spot. So it acts as a way to deter EcoR1 from cutting up its own genome. It's essentially this methylation protects the bacterium from cutting up its own genome. So this methylation occurs by an enzyme called a methyltransferase that puts this methyl group onto some of the adenines in the system. So there are many, many different restriction enzymes. If you go back to video part 1, we talked about plasmids and the multiple cloning site. There's a bunch of examples in there of the different restriction enzymes. We looked at some in the lab. Restriction enzymes usually run from say 4 to about 12 base pairs. And they can make sticky ends. They can also make blunt ends. This is where they cut. Let's just pretend this is a different sequence. They cut straight up and down. And so those ones cannot be used to put your DNA back together. The shorter sequences are going to be less specific, so they're going to cut more times within a genome. The longer the sequence is, the more specific it's going to be, the less times it's going to cut in a genome. The interesting thing about laboratory application of restriction enzymes is that a particular genome cut with a specific restriction enzyme is always going to make the same pattern. So this is the basis for DNA fingerprinting, which is used in crime scene investigation. It's used in paternity testing, basically to identify a person's unique DNA sequence. And so if you cut a genome or a portion of a genome where you expect there to be a wide variety among individuals, you can get specific, unique DNA fingerprinting patterns. So finally, we get to our non-sequence specific or sequence non-specific defense mechanism. So CRISPR and restriction endonucleases are both sequence specific. But bacteria can also cut up or degrade sequences without being specific, pieces of DNA without being sequences specific. Excuse me. One is by recognizing linear DNA. So the bacterial chromosome is going to be circular. The genomic chromosome is going to be circular, as are any plasmids. So if we have a plasmid in here, it's going to be circular. And so if the bacteria is that infects the cell is linear, the cell is going to recognize this as foreign. Now, linear DNA, especially if it happens to be single-stranded, is very unstable, and the cell will break it down really easily. So if a piece of linear DNA is incorporated and it doesn't become circular for some reason right away, it's going to be broken up into little pieces so that it cannot infect or be maintained in the cell. Second, we can repress transcription of the sequence. So this is a system that's not necessarily breaking down our DNA that's come into the cell, but it is keeping it from being transcribed. So most bacterial genomes are GC rich. So cells can identify or recognize bacteria that is not GC rich. In other words, it can identify DNA that is AT rich. So let's say that you have a virus that has infected the cell that is AT rich. That cell can recognize the AT rich regions and use a specific enzyme to bind and block transcription. So we're not breaking it down, but we're keeping it from being transcribed. So in E. coli, this enzyme is called HNS. And HNS binds and silences or blocks those AT-rich regions so that they're not transcribed. But the normal DNA sequences, those GC-rich sequences, are going to continue to be transcribed regularly. That is to say that the normal genomic DNA is going to be transcribed as usual. But any horizontally acquired DNA from, say, a virus or a bacterium in the environment is not going to be transcribed. So this sort of blocking of DNA or breakdown of DNA from an outside source also occurs in humans, and the antiviral activity in humans can actually prevent DNA of infecting viruses from infecting our cell or taking over our cells. So a particular group of viruses called retroviruses insert RNA into the cell. So they infect a cell with RNA. HIV is a retrovirus, and HIV infects RNA into the cell. And when it gets into the cell, the RNA is copied into DNA, and it eventually makes a RNA to RNA DNA combo, and eventually makes a DNA combo. So these retroviruses infect with RNA, but eventually is turned into DNA. So there's this special enzyme in mammalian cells that recognize these unique RNA DNA hybrids. So reverse transcriptase is going to copy that RNA into DNA and make a double-stranded molecule. Now, this ApoBec3G enzyme has a specific job that recognizes these DNA RNA hybrids and acts as a mechanism to block infection. So when it blocks infection, it recognizes the cytosine in the RNA molecule. So if this black sequence here is our RNA molecule, it's going to recognize the cytosines in that RNA DNA hybrid that are in the RNA molecule. I'm sorry, let me back up. This is our RNA. Let me get the right one. It is going to recognize the DNA complements cytosines. And when it recognizes the DNA complements cytosines, it's going to deaminate or take off an amine group. This is one of an amine group from the nitrogenous base. And so it is going to, the nitrogenous base, the part that sticks off the sugar. It is going to take an amine group off of that cytosine and turn the DNA into a U. So we've got our RNA here. It's going to incorporate or change that cytosine into a U. It's not going to incorporate. It's going to change that cytosine into a U. So then when that DNA is replicated, and this is our DNA strand, even though it's got a U, it is going to copy that U into the complement, which is an A. So where our original virus sequence had a G, we now have A's. And so it has replaced the U that was incorporated, turned it into an A on the complement, and then introduced, when it is replicated again, introduced a T. And so basically you get this flip-flop of nitrogenous bases that now have mutations. And this is going to basically be nonsense to the cell. It's not going to code anything anymore because the original RNA sequence had a C there, and now it has a T in the DNA that has been infected into the cell. And so that DNA that is now full of mutations cannot properly produce new viruses. So we get no new viruses, which means that viral infection is not going to spread to other cells. Now, this is not necessarily all cells, and it doesn't mean that it's going to block all viral infections, but it's definitely going to slow down viral infection in some cells.